In today's video, we'll discuss about prokaryotic cell. In the last session, we have gone through the introduction of cell, and today we'll go with prokaryotic cell. Now, if you talk about prokaryotes, the word prokaryote and eukaryote that was derived from a Greek terms, where pro that indicates before, karyon that means nucleus. It means these members are formed before formation of nucleus. So they are considered as prokaryotes. There are different members that come under prokaryotes. These members that includes bacteria and mycoplasma, blue-green algal members, PGA you can write, and it also includes purple and green sulfur bacteria. Green sulfur bacteria. Now here, especially, right, there is a kingdom which particularly consists of prokaryotes only, and that kingdom is Monera. In Monera, it includes all prokaryotes, and in those prokaryotes, particularly bacteria are considered as the major members or sole members you can say. So here bacteria are the sole members of Monera. Now these prokaryotes, they look very simple, but they perform different life functions, due to which we can call them as simple in structure and complex in behavior or functions. Okay, now there are different types of prokaryotes are present. Basically, they are of 4 types. Now, if you look at these 4 types, it can be rod-shaped, which is also be called as bacillus. It can be spherical, which is coccus, comma-shaped, vibrio, and spiral-shaped, which is spirillum. So these are some of the types of prokaryotic cell structures based on their shape. Now, let's go with the structure of prokaryote. Structure. Now, if you look at the structure, they are basically covered by cell envelope, which is protective in function. So we'll talk about it. So here, cell envelope. Number 1, cell envelope. Now, under cell envelope, we have to focus 3 different things. Number 1, glycocalyx. Number 2, cell wall. Number 3, plasma membrane. The outermost covering, or is the outermost part of this cell envelope, that includes glycocalyx. So here you can see we are taking glycocalyx. Now this glycocalyx, it can be a loose sheet or it can be thick and tough. So what can it be? Number 1, it is glycocalyx. Now this glycocalyx, it can be a loose sheet. If it's a loose sheet, it is called slime layer. Now this slime layer that helps in preventing from loss of water, or else this can be thick and tough layer. If it is thick and tough, that is called capsule. Now here the function of capsule is important. It hides the bacteria from host immunity. So what does it do? Right? It hides bacteria from host immunity. Host immunity. So this is a function of capsule. Now this is the outer covering. So what is that? It's a glycocalyx. Here you can see glycocalyx, outer covering. Number 2, inside to this glycocalyx, there is a thick rigid layer is present, and this thick layer, it is called cell wall. And this cell wall, it is made of amino acids and sugars. So here, this is a cell wall. Number B, cell wall. Now, if you talk about this cell wall, it is mostly present. There is an exception is also there in the prokaryotes which does not consist of cell wall, that is mycoplasma. So if a question is given, out of the following which is a wall-less monera or wall-less prokaryote, then the answer is mycoplasma. So here cell wall, it is dead, rigid, right? And it is mostly present. Mostly it is present. Now, cell wall, it is also the basis for the classification of gram-positive and gram-negative bacteria. So here, this becomes the major criteria in the gram staining technique. If it is composed of only peptidoglycan layer, now that gives a positive effect for gram staining, so called as gram-positive bacteria. Now this peptidoglycan layer, if it is hidden by lipopolysaccharide or else LPS layer, that gives a negative effect to the gram staining, due to which it is called as gram-negative bacteria. As NCERT does not talk much about this description, so we are keeping it limited. Now whatever this cell wall is there, it determines the shape of the cell and helps in protecting the cell from collapsing. Now, it is made of what? Peptidoglycan. Peptidoglycan. We can also call it as murin or mucopeptide also. Now, if you talk about peptidoglycan layer, in the name itself, right, you can find two terms, peptide and glycan, where peptide that indicates protein or else amino acid, glycan that indicates sugar. Below the cell wall, it consists of plasma membrane. Now this plasma membrane, it consists of infoldings, and these infoldings can be of vesicular infoldings or tubular infoldings, or else they can be lamellar infoldings like this. This is a vesicular infolding, this is a tubular infolding, this is a lamellar infolding. Number C, plasma membrane. Now if you talk about plasma membrane, it is selectively permeable in nature. Selectively permeable in nature. It is present below cell wall. It consists of infoldings, and these infoldings, they are usually present in only some gram-positive bacteria. It means presence of mesosome. It's a characteristic feature of only some gram-positive bacteria. Now these infoldings, they are called as mesosome. I already said these are of three types. They can be vesicular infoldings. Number 2, tubular infoldings. Number 3, lamellar infoldings. Now, what is the function of these mesosomes? Now, if we talk about the function of these mesosomes, they help in formation of cell wall. They help in formation of cell wall. Number 2, they help in DNA replication. Number 3, they help in respiration. Especially, these act like a site for the electron transport system for the production of ATP. And secretion. And as it is a infolding, it increases the surface area of absorption and enzyme action. So what it do? It increase absorption area and enzyme action. Enzyme action. So these are the functions of mesosome. Now here you might be getting a doubt. Sir, in majority of the members, mesosome is absent. Then who is going to replace the functions of mesosome? So you have to remember a point. Mesosome is a infolding of plasma membrane. It means it's also a function of plasma membrane. So these all functions, if mesosomes are absent, that will be done by plasma membrane. Number 2, that is genetic material. Genetic material. Now, if you talk about the genetic material of prokaryote, it is called as genophore. Genophore, geno, that means gene, phore means thread. It's a gene bearing thread. And the packed form of this genophore is called as nucleoid. So what is the packed form of genetic material in prokaryote called? Nucleoid. So generally it is called genophore. Packed form is called as nucleoid. Now whatever this nucleoid is there, it's a double-stranded circular DNA. Double-stranded circular DNA. So if a question is given, presence of double-stranded circular DNA is a characteristic feature of which members? Then you can take the property of prokaryote as an answer. Now, whatever this nucleoid is there, it is composed of three things. Number one, that is DNA, that's already over. Number two, it consists of RNA. And number three, it consists of non-histone proteins, or else non-histone amino acid you can say. Now whatever these non-histones are there, they are basic amino acids, which makes it different from eukaryotes. In the eukaryotes, the non-histones are acidic. In the prokaryote, the non-histones are basic in nature. Now, so here I am taking the point. So there's the double-stranded circular DNA, which is called as nucleoid. Now, apart from this one, there is an extra genetic material is also present, and this extra genetic material, it is called as plasmid. And this plasmid, it is also a small double-stranded circular DNA. Now, what is the function of this plasmid? If you talk about a simple function of this plasmid, it provides unique property or unique phenotypic character to the bacteria. So it provides unique character to bacteria. So what we have completed now? Number one, cell envelope. Under cell envelope, glycocalyx. Number two, cell wall. And number three, plasma membrane we have completed. Now these three things, they come under cell envelope system. If a question is given, what is the overall function of cell envelope system? What is the answer? It is protective in function.